So today I'm going to talk to you guys about the subject of peptides and ACE inhibitors. So there are some naturally occurring peptides that can function as potent ACE inhibitors and help to control blood pressure. So we're going to go over what exactly makes these peptides effective ACE inhibitors and why we use a very particular one in our product HemoFlow. So first off, ACE stands for angiotensin converting enzyme. It converts angiotensin I, which is a protein, into angiotensin II. Now, what angiotensin II does in the body is it causes the narrowing or constricting of blood vessels, also increases sodium and water retention, and stimulates the release of aldosterone. All of these things can lead to increased blood pressure. Now, what an ACE inhibitor does is it prevents the angiotensin converting enzyme from producing angiotensin II by inhibiting that enzyme. So there are several ACE inhibiting prescription medications that you can take that you can be prescribed if you have blood pressure issues. So why are we looking at naturally occurring ACE inhibitors instead? Well, some people have difficulty going through the whole process, finding a competent doctor who's going to be willing to prescribe medication to them. It could be cost issues, not having proper insurance to be able to afford medication, this sort of thing. And also, medications that are prescribed for blood pressure can have some undesirable side effects like headaches, dry cough, and edema, creating demand for some more natural sources of ACE inhibitors. So let's take a look at ACE inhibitors and peptides from food origins. So ACE inhibitory activities of peptides can be affected by their structural characteristics, such as chain length, composition, and sequence of these amino acids to form a peptide chain. Another important thing is that ACE inhibitory peptides must have gastrointestinal stability, and they must be able to reach the cardiovascular system in order to show their activity. Now, a lot of peptides that we eat in our diet do not have gastrointestinal stability, and they are broken down into their amino acid structures during digestion. Now, what many of these studies for ACE inhibitory peptides have found is that shorter peptides, which are di or tripeptides, meaning 2 or 3 amino acid sequence, are more effective and they're more permeable across small intestinal epithelia. Now, what many of these studies show is that the presence of the amino acid proline at the C terminal end of the tripeptide. So if you remember, peptides contain both an N terminal and a C terminal. And some examples of the tripeptides we'll be looking at in particular today, like LKP, IPP, VPP. These are all abbreviations for the peptide structure. Each letter represents one single amino acid, but you can see what all three of these have in common. They all have the P at the end, which stands for proline, and proline is at that C terminal of this tripeptide complex. So having the presence of this proline at the C terminal of the tripeptide competitively inhibits ACE activity. The exact reason and mechanism isn't quite known, but it's probably some sort of combination of having GI stability, having a short length of the chain of the peptide sequence, hydrophobicity, and various other molecular and electronic properties that allow this peptide to interact with its environment properly to be able to orient itself to be an effective ACE inhibitor with that enzyme. So particularly with tripeptides, research shows 3 pretty effective tripeptides for their ACE inhibiting activity. And those 3 that we're going to look at are LKP, VPP, and IPP. So let's just take a look at each of the data on these. LKP is actually the most effective ACE inhibiting peptide found in what's known as bonito fish peptides. So bonito fish peptides have had some research showing their effectiveness on blood pressure in animal studies. Basically, the 2 effective peptides in here are LKP and another 5 amino acid sequence peptide that contains LKP as well. But since LKP is the shortest one of these, it's the most effective. So that's kind of where we focus on. So when it comes to bonito fish peptides, there's no actual human studies done on it. It's only rat or other animals. Some clinical studies on sardine dipeptides, which amounted to a systolic reduction of 9.7 millimeters of mercury and diastolic by 5.3 millimeters of mercury. So that's just kind of a comparison that we can use when we compare to VPP and IPP. So these VPP and IPP peptides are actually lactotripeptides. That means they're found in milk proteins. So a study with VPP and IPP showed a reduction of 13 millimeters of mercury in systolic and 8 millimeters of mercury in diastolic. And this is referring to a human study. Now, this is the most significant reduction of blood pressure using peptides in humans compared to any other sort of peptide-based study. Again, the peptides were studied include fish, milk, legume-based, all sorts of different dietary peptides. So why is LKP a less efficient ACE inhibitor compared to VPP and IPP? Well, we have the structures here, and we can just take a look at all three of these structures. We can predict that LKP is probably less effective just because it is a larger and bulkier molecule, making its ability to interact with the angiotensin converting enzyme a little bit more difficult. So here we have LKP. Note that these are not really to scale, but you can see a couple long chains here. And then we have the structures of IPP and VPP. They are just a little bit more condensed. The chains aren't quite as long. So that can give us a good estimate of why VPP and IPP are superior in the studies that we have in humans. Now let's talk about the effects. So blood pressure lowering effects are typically measured 4 to 6 weeks into treatment, but sometimes you can notice a difference after a week or two. Now in these studies that led to the 13 and 8 millimeters of mercury reduction, a total of 3.4 milligrams of VPP and IPP combined as a dose was used. Now that number is important to note because that is the basically exact equivalent of 508 milligrams of VasoDrive-AP. Now VasoDrive-AP is the ingredient that we use in our product HemoFlow, and it contains these two lactotripeptides, IPP and VPP. VasoDrive-AP is used most commonly as a sort of pump product because of its vasodilation properties. Well, the reason it has these vasodilating properties is because of the topics I discussed above. So it's used for vasodilation and an ACE inhibitor for blood pressure due to both of these lactotripeptides, IPP and VPP. Now, there are plenty of studies looking into antihypertensive peptides, and out of all these, IPP and VPP have the most clinical data, and they show the most drastic results in humans with only as little as 3 milligrams or 3.4 milligrams figure of combined peptides per dose. Now, VasoDrive-AP has substantiated claims for healthy blood flow and maintains healthy blood pressure already in the normal range. Additional benefits of these two lactotripeptides found in VasoDrive-AP can be used for training is that it has synergistic action with nitric oxide or enos ingredients that are commonly found in pump products that provide active vasodilation, while VasoDrive will basically amplify and sustain that vasodilation by preventing the vasoconstriction coming from angiotensin II from taking place. So again, the effects during training on this is that this ingredient can help prevent vasoconstriction on blood flow during training. And some of the other benefits it provides is it improves exercise capacity, blood flow, which we've talked a lot about blood flow and those interactions with slower moving red blood cells that have the potential to clump together, recovery from intense muscle damaging exercise, and reducing delayed onset muscle soreness. So again, this is just a quick summary of what we've learned from these ACE inhibiting peptides that we can find in natural foods. Things like VPP and IPP can have a very substantial effect on blood pressure and might just be more convenient, might be safer to use, or just cheaper whenever it comes to comparing this to a prescription ACE inhibiting medication.